Welcome children. Today we are going to start a new chapter, Biomolecules. This is the chapter 9 of class 11th biology. Children, I would like to say you that this chapter is very important, not only for examination point of view. The understanding of this chapter is quite helpful for understanding of some of the chapters of class 12. So, I request all of you to listen to me in this video carefully and note down the points. Okay, so let me first tell you what we are going to discuss. So in this chapter we will be discussing the biomolecules, or the molecules that is present, the chemical compounds present in the living body. Okay, and that includes protein, carbohydrate, lipid, nucleic acid. Then that we'll discuss about the enzymes. So today in this session we'll be discussing biomolecules, introduction, and then proteins. So let us start our discussion. So children, we observe the elements present in the non-living and living matter are quite similar. Only their quantity may vary. So many researches have been taken place by different scientists, and it was observed that whatever elements are present in the earth crust are also present in the human body, and only they vary in their quantity. So here in this chart, I have given you some of the elements which are present in both Earth's crust and human body. So here you can see hydrogen. It is present in both human body and Earth crust, but the percentage weight is different. In Earth crust it is 0.14, but in human body it is 0.5. The carbon element is present in the earth crust in the atmosphere, that is 0.03, and in human body it is 18.5. Similarly, oxygen, its weight percentage in earth crust is 46.6, but in human body it is 65.0. Nitrogen, very little amount present in the earth crust, but in human body it is 3.3 percentage weight. Sulfur 0.03 in earth crust, and in human body it is 0.3. Sodium 2.8 in earth's crust, in human body it is 0.2. Calcium, it is 3.6 in earth's crust, and in human body it is 1.5. Magnesium, in earth crust it is 2.1, and in human body it is 0.1. Silicon, in earth's crust it is 27.7, and human body it is negligible. It is 0. Okay, you can say. Now, how to analyze the chemical composition of a living system, whether it may be a plant or animal? How to know the presence of chemical composition? So these chemical composition may be organic or inorganic. So first we will discuss how to analyze the organic chemical composition in a living system. Okay, so this is the description of analysis of organic chemical composition in a living system. Okay, so for that purpose we have to follow following steps. So what are those steps? We have to take any living tissue, which may be a vegetable or a piece of liver of any animal. Then we have to grind it in trichloroacetic acid. Very important, children. So this question is asked in the exam. Which chemical is taken for analysis of organic chemical composition in living system. So it is trichloroacetic. So this can be ground by using a mortar and pestle. Okay, and so that we will get a thick slurry. Okay, then we have to strain this through a cheesecloth or cotton so that we'll get two fractions. Okay, and one is called filtrate, and that is more technically the filtrate is the acid soluble pool, and the second one is the retenant or the acid insoluble fraction. So when we strain it, whatever remains in the cotton or cheesecloth, that is the acid insoluble fraction, and the fluid part, that is the filtrate, is the acid soluble pool. Okay, so it was observed that in the acid soluble pool, thousands of organic compounds were found. So this is how the scientists analyzed the presence of organic compounds in the living system. Okay, so where do you find the organic compounds? It is present in the acid soluble pool. Okay, very important, children. So the organic compounds are present in the acid soluble pool. So these are the steps. So living tissue is taken. You can see the steps whatever I have already discussed. It is grind in the trichloroacetic acid. Then we got a thick slurry. Then it is strained through cheesecloth. Then we got two fractions. One is filtrate, which is the acid soluble pool, and the retenant that remains on the cotton cloth, and that is the acid insoluble fraction or the pellet. So in exam, if this process is asked, you can represent the process with the help of this schematic diagram. You will get full mark. Now, how to analyze inorganic compositions? So for this purpose, what we have to do, it is a bit destructive process, but to analyze, it is conducted. So when one weighs a small amount of living tissue, when we take living tissue, then it is called weight. Okay, so because the fluid is there and it is weight mass, so it is called weight of that tissue. Then we have to dry it. So how? When the water will evaporate from this tissue, then that will be known as dry weight of the tissue. Now, if the tissue is fully burnt, all the carbon dioxide, all the carbon compounds. So carbon compound means organic compounds. Organic compound will get oxidized to form gaseous form of carbon dioxide, water vapor, and they will be removed. Is it clear? So first we have to take the tissue, we have to dry it, then we have to burn it. So when we will burn it, the carbon compounds, means all organic compounds, will get oxidized and it will be converted into either gaseous form or water vapor, and they will be removed. Then what will be remaining? The remaining part is the ash. And the ash contains inorganic elements like calcium, magnesium, sodium. So these are the inorganics. Now we can separate the inorganic materials or inorganic matters from the organic matter. Okay, and besides that, we can also get the inorganic compounds like sulfate, phosphate in the acid soluble fraction, that is that we have discussed in the previous process. In previous process, we obtained two fractions, that is acid soluble and acid insoluble. So in acid soluble, basically we are getting the organic compounds, but along with that, we can also get sulfate, phosphate, etc. So some inorganic compound may also be present in the acid soluble fraction. Okay, so therefore the elemental analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. That is, we'll get the elementary form. Okay. So now we'll discuss what is biomolecule. The chemicals or molecules present in the living organism are known as biomolecules. And these biomolecules are of two types. As I have said you, they are inorganic and organic. But in your textbook, it is given that the organic compounds found in the living system are called biomolecules. Okay. So, for examination, you have to write that definition. But actually, these are also biomolecules. The inorganic molecules are the biomolecules. So, both are biomolecules. The inorganic biomolecule includes the minerals, gases, water, and the organic molecule includes carbohydrate, fat, protein, nucleic acid, vitamins, etc. And the different biomolecules, like the organic, I'm talking about the organic biomolecules, can be classified as aldehyde, ketones, aromatic compounds as the chemical forms. Okay. And what are the chemical forms? The chemical forms are amino acids, nucleotides, fatty acids, and they are known as the biochemical forms. So here let us see what are biomolecules, the classification of biomolecule. This classification is very, very important, children. So the biomolecules are of two types, that is micromolecules and macromolecules. Okay. So what are the micromolecules? The micromolecules are the small sized molecules which are having low molecular weight, and their weight is between 18 and 1800 daltons, and which are found in the acid soluble pool. Okay. So in the acid soluble pool, that is the first process of analysis of chemical composition which I described. In the filtrate, we will be getting the low molecular weight molecules whose molecular weight is maximum up to 800 Dalton. So what are those molecules? They are minerals, gases, water, sugars, amino acids and nucleotides. Now what are the macromolecules? The macromolecules are the large sized high molecular weight whose molecular weight is above 10,000 Dalton. So these molecular weight you have to remember, children. On the basis of this molecular weight only they are classified as macromolecule and micromolecule, and they are found in the acid insoluble pool. That means the pellet that remains on the cotton or that is the cheese cloth. Okay. So what are those compounds? Those compounds are carbohydrate, lipid, protein, nucleic acids. So these are the macromolecules. Okay. So actually the lipids are the micromolecules, but since the lipid always remain in a globular form, in aggregate form, so its molecular weight is more than 10,000 Dalton. So it is categorized as a macromolecule. This is one of the most important questions that comes in the exam. So this is a list of representative inorganic constituents of living system. Sodium, the formula are given. So sodium, potassium, calcium, magnesium, water, and compounds like NaCl, CaCO3, phosphate, sulfate. So these are the inorganic constituents present in the living system. So here, this point you have to note down, children. Except lipids, macromolecules are formed by polymerization of subunits called monomers. So, what is polymer? That you know that you have studied in class 8th, the synthetic fibers. In that chapter, you have studied about the polymers. So, poly means many, mer means units. So, many units, single units, when they are joined together to form a chain, a straight chain or a branched chain, then that is called polymer, and the process is called polymerization, and the single unit is called monomer. So now what I said, except lipid, lipid consists of two things. So I will be discussing while dealing with lipids. So all other macromolecules like protein, carbohydrate and nucleic acid, they are all polymers. So let us see what are the polymers and what are the monomers of each polymer. So here you can see that the monomer, that is simple sugar or the monosaccharide, form a polymer that is called polysaccharide. And for example, starch, cellulose, glycogen in animals, and that is that they can be isolated, they can be determined, their presence can be determined by using the reagent Benedict, glucose, iodine, starch. So these are the practical part. So still then I am telling you because it is there in your practical. Okay, so this table is very important, children. So for polysaccharide, what are the monomer? Monomer are the simple sugar or monosaccharides. Then amino acid are the monomer which combine together to form a polymer that is called protein or polypeptide. They are present in hair, enzyme, hemoglobin, insulin. Okay, and they can be tested by, their presence can be tested by using the reagents like ninhydrin biurate test. Then the next one is nucleotides, are the monomers which combine together to form the nucleic acid, that is DNA and RNA, and they can be determined by, they can be identified by presence by using the reagent methyl green. Then fatty acid and glycerol are the, so why I told that except lipid, because in lipid you can found two monomers are there. So they are combined together, and this fatty acid may vary. Okay, so they are the monomers which form the fat or the triglycerides, and they are present in the cooking oil, butter, etc., and they can be identified by using grease test with brown paper. So this table is very important, children, to know the monomers for the polymers along with their examples. Now we will discuss the important polymers, that is biomolecules, that is in your text, that is in your syllabus one by one. Okay, so let us start with proteins. So what are proteins? Proteins are the polymers of amino acid, and the protein composed of the elements carbon, hydrogen, oxygen and nitrogen. Very important, children, you have to remember. Protein consists of the element carbon, hydrogen, oxygen and nitrogen. So here you can see the structure of the amino acid, the monomer, and how this monomer forms a polymer, that also I will show you. So here you can see this is the structure of a single amino acid, only this portion. Okay, so here you can see that centrally placed carbon atom is there, and you know that carbon has 4 valency. So to carbon is attached to this carbon, to one end hydrogen is attached, then to one end carboxyl group COOH group is attached, to another end the amino group is attached, NH3 group is attached. So why it is known as amino acid? Because this is a macromolecule which contains both amino group and acid group. That's why it is called amino acid. Now what about the R? R is the alkyl group that can vary from amino acid to amino acid. So in all the amino acid, we will see that these three structures are same, that is amino group, hydrogen and carboxyl. All these three are same. But what they are different from each other? They are different in R. That means only this part is different from each other. So here, in order to show the variation, I have shown you the 3 amino acids, how they are different in their R. So the simplest amino acid is glycine. Remember children, very important. So glycine, as I have said you, one is acid group, one is amino group, one is hydrogen. So these 3 are constant, and the R, in place of R, what is there? Hydrogen is there. So then it is called glycine. Then in alanine, you will see that these 3 are common and R is CH3. Okay. Then in serine, you can see that these 3 are common and what is different? CH2OH. R is different. So in this way, the amino acid are different from each other in the variant group, that is R group. I think it is clear to you. Now I will tell you how the polymer is formed. The amino acids are joined together to form a polymer. Remember children, the amino acids are joined with one another by forming a special type of bond, that is called peptide bond. Very, very important children, in exam the formation of peptide bond is asked as a 2-mark or 3-mark question. Okay, so you have to show the peptide bond. Now let me explain you in details. So here this is one amino acid. So as I have told you, these 3 are common. Okay, so COOH, then H, then NH2. These 3 are common. Listen to me carefully children. And here R1. So this may be any group because I have not named the amino acid. This is amino acid 1. Then in amino acid 2, these 3 group are same but R2. That means this variant group is different. Now, in an amino acid, at this end, N is present. Amino group is present. So this is called N terminus. And this is called C terminus because at this end carbon is present. Okay. So this also you have to remember. Now what happens? These 2 amino acids join together to form a polymer by a peptide bond. Now how it takes place? You just see children. So this is one amino acid. Okay. This is another amino acid. So here what was there? OH was there. Okay. So here OH was there and here what was there? Here what was there? With N, H was there. Again I am repeating. With this amino acid, just see, with this C, what else was there? OH was there. Okay. And with this N, what else was there? H was there. Now what happens? During the formation of peptide bond, these OH and H, this highlighted portion, just see children. So this is removed as water. Okay, so here you can see the water is removed and the peptide bond is formed. Okay, so this bond CONH bond is called peptide bond. So in this way, the amino acids are joined with one another by peptide bond to form a protein chain or polypeptide chain. Okay, so since here 2 amino acids are there, then it is dipeptide. Similarly, 3 amino acid, then it will be tripeptide, tetrapeptide, so on. Okay, so I think this process is clear to you. If you have doubt children, you can pause the video and listen to me carefully. Okay, so this is how with the removal of water molecule, the peptide bond is formed and protein is formed. Now we'll study about different types of amino acids. The amino acids are basically of 2 types, essential amino acids and non-essential amino acids. What are essential amino acids? The essential amino acids, see in the living system there are 20 types of amino acids. out of which some amino acids cannot be synthesized in human body. So these amino acids are required to be taken in diet. Okay, so they are known as essential amino acids. But there are certain amino acids which are synthesized in our body. So they are not required in our diet. So they are known as non-essential amino acid. Besides these two type of amino acid, there are some other type of amino acids are also there that we will discuss later on. So now these are the examples of essential amino acids that we obtain from the nutrition. Okay, they are leucine, isoleucine, valine, histidine, lysine, methionine, phenylalanine, threonine and tryptophan are the essential which we have to take in our diet. Then the amino acids which are synthesized in our body are the non-essential amino acid and they are alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine and tyrosine. So children, you have to remember some examples of essential and non-essential amino acid because you sometimes it asks in the exam, give some example of essential and non-essential amino acid. So now some other classification of amino acid. Basing on the chemical structure, the amino acids are polar amino acid and non-polar amino acid. Okay, I will show you what is polar amino acid, what is non-polar amino acid. So the polar amino acid may be neutral or charged. Non-polar amino acid may be aliphatic, aromatic or sulfur containing amino acid. So you know that aliphatic means open chain, the R will be having open chain and the aromatic will be having aromatic ring in place of R. Then sulfur containing, that is in the side chain, that is R, the sulfur will be there. So now here you can see the polar and non-polar amino acid. Here some example I have given. So here you can see cysteine is one example of polar amino acid where you can see that this is the R and this shows polarity. This shows polarity. Okay, and but here you can see that this is leucine is non-polar because the R is non-polar. Okay, the variant group is non-polar. Okay, similarly the charge, depending upon charge, here you can see that negatively charged or acidic, the negatively charged amino acids are the acidic amino acid and positively charged amino acids are the basic amino acid. So from the structure I will make you clear that here these three are common in both the amino acid, but in the side chain, that is in variant group, what do you find? Another extra negatively charged compound or extra acid group. So it is called negatively charged amino acid or acidic amino acid. Extra acid group is there. Similarly here these three are common, but in the side chain what is there? Amino group is there. It is a basic group, so it is basic amino acid, and NH3 is positively charged, so it is positively charged amino acid. So children, you have to understand these concepts very clearly because in class 12, while dealing with molecular basis of inheritance, again you have to, I mean you have to utilize these concepts in that particular chapter. So please children go through this, okay, very seriously. Now these are the list I have already told you, no need to repeat. Then one of the most important feature of amino acid is that they can exist as both acidic or negative ion or positive ion, or only acidic or only basic ion. Okay, so this happens when the pH changes. So based on the number of amino and carboxyl group, they are acidic and basic, as I have shown you in the previous picture. Okay, and neutral one is valine. Valine is neutral. Remember, this is very important. So in previous one, I have shown you the acidic and basic one. But you have to remember that valine is neutral amino acid. Okay. So neutral means that is both positively and negatively charged. So how? Let me show you here. This is positively charged and this is negatively charged. Okay. So here you can see. Okay. But here you can see that this one is neutral one. Okay. So here you can see that this is converted into this is having both positive and negative charge. This will be very clear in this diagram. Let me show you. So here you can see that this is at low pH. When the pH is less than 3, when the pH is less than 3, then the amino acid is in this ionic form, that is positively charged. Okay, so that means they are basic. So when the pH is more than 10, they are acidic. They are acidic or negatively charged. But at neutral pH, when the pH is neutral, that means 7, then they are having both positive and negative charge in equal amount. So this is called deuterion. This property is called deuterion. Deuterion means the amino acid can exist as both positive and negative ion, act as both acid or basic, that is you can say neutral at neutral pH. So it is called deuterion. This is also very important, children, from examination point of view. Now here we'll discuss some important function of the proteins in living cells. Collagen, intercellular ground substance. It forms ground substance. You know that it is a part of connective tissue. Trypsin is the enzyme which helps in digestion of protein. Insulin is a hormone that controls the blood sugar. Antibody, you know, these are also protein. They fight the infectious agents. Receptors, they are present in the cell membrane and as well as in our tongue. They are present as the receptors and they sense various stimuli. Then glutamate 4, it helps in transportation of the glucose into the cell. Besides this, some other functions are there that we will discuss at the end. But the most important thing, children, you have to remember, I have highlighted here, collagen is the most abundant protein in animal world. 1 mark question. Ribulose biphosphate carboxylase oxygenase Rubisco is the most abundant protein in the whole of the biosphere. 1 mark question. So these two often come as your 1 mark question. So please, children, be serious. Then we'll discuss the structure of protein. We have discussed the structure of amino acid. Then we discussed how the amino acid join together to form the peptide chain, how the peptide bond is formed, that we discussed. Now, the protein has also different levels of structure. So, what are those structures? The level of structures are primary, secondary, tertiary and quaternary. Now, in different level of structures of protein, different types of bonds are involved. Very, very important, children. You have to remember, I am giving you in a very systematic way. Okay. In your textbook, it is not given systematically, but I have made it systematic. So, you can make note from this one also. That will be very helpful for you. So, primary structure, in primary structure of protein, the peptide bond is only peptide bond is present. Peptide bond is a covalent bond. You must be knowing what is covalent bond, what is electrovalent bond. So I am not talking much about those types of bond. So peptide bond is a type of covalent bond and it is present in the primary. It is the only bond that is present in the primary structure. Then in the secondary structure, hydrogen bond along with peptide bond is present. Along with peptide bond is common. in all the 4 level of structure. Along with the peptide bond, hydrogen bond is present between the amino acid. Then in tertiary structure, along with peptide bond, hydrogen bond, ionic bond, disulfide bond, hydrophobic interaction are present between the R group. Okay, so these are the different types of bond. So see how the number of and types of bond is increasing as the structural level is increasing, and that's why the structural level becomes more and more complicated. Okay, then quaternary structure here in between the polypeptide chain. So here the amino acids are not bound together. Here the polypeptide chains are bound together with the help of hydrogen and ionic bond. So let us discuss different structural level of protein one by one. So this is the same thing, but here I have shown you the structure. So here you can see this is the simplest structure, primary structure, very simple. Then secondary structure is bit complicated. Then tertiary structure is more complicated, and quaternary structure is the most complicated structure, which is the combination of more than one polypeptide chain. The same thing, but the description is there. Okay, so primary protein structure consists of only the sequence of amino acid joined by peptide bond. Then the secondary protein structures are of two types. One is beta pleated sheet and other one is alpha helix. So in one structure, the protein chain may form alpha helix helical structure, or it may form a folded paper like structure, that is called beta pleated sheet. Then you can see that tertiary protein, it is the combination of beta pleated and alpha helix. So in secondary structure, you will see either alpha helix or beta pleated sheet separately. But in like a tertiary protein structure, it is the combination of beta pleated and alpha helix structure, and that's why it is more complex. Okay. Then in quaternary protein, you can see that is here I have shown you two different polypeptides are joined together. Now let us discuss the primary structure of protein. The primary structure of protein is the basic structure. Here a number of polypeptides are involved, that is amino acids are involved. The first amino acid is called N-terminal amino acid. From the left side, the first amino acid is called N-terminal and the last amino acid is called C-terminal. Very, very important, children, because I have shown you that in the amino acid on the left side, the amino group is there. So, N is there. So, it is N-terminal. And at the end of the protein, which end will be there? The carboxyl group will be there. So, that will be C-terminal. So, here let me show you. So, this is the primary structure of a protein where simply the amino acids are joined together to form a chain. But sometimes here most important thing I am going to tell you, which comes in the exam, that is insulin. See, the proteins are inactive in their primary structure. All proteins are inactive in their primary structure except insulin. So on the right side in this box, I have given you the structure of insulin. So here you can see that insulin composed of actually it is composed of three chains, that is A, B, C. But in the active insulin, the C chain is removed and only A and B chains are there. So here C chain is not shown because it is active insulin. So here you can see that this is a polypeptide chain and this is another polypeptide chain, and they are having the that is the B chain and the A chain. So they are having primary structure. So these primary structures are simply joined with one another by the formation of disulfide bond. But the whole structure is a primary structure only. because here no hydrogen bond is formed. So we cannot say that it is secondary structure. So it is primary structure of insulin, and insulin is the only protein which is active in its primary structure. Very, very important, children. It is that's why I have highlighted this particular portion. So this is insulin. I am repeatedly, because I have not labeled this as insulin. So this is the structure of insulin protein, which consists of A chain and B chain. This is the primary structure of protein, and this is the only protein which is active or functional in primary structure. Then come to secondary structure of protein. So secondary structure, as I have told you, they form alpha helix or beta pleated. Okay. So the alpha helix peptide chain is coiled spirally in right-handed manner, and beta pleated protein, that is in which the two or more peptide chains are interconnected by hydrogen bond. So in secondary structure, presence of hydrogen bond is must. And then another type of secondary structure is the collagen, where three strands of polypeptides are coiled around. So in alpha helix only one strand is there. In beta pleated two polypeptides are joined, and in collagen three polypeptides are joined, and all of them are joined by hydrogen bond. This is very important, children. So here you can see this is alpha helix, where a single chain is folded upon itself, and you can see that this dotted structure shows the hydrogen bond between the amino acid. So this is one amino acid, this is another amino acid. In between the first and third amino acid, the hydrogen bond is formed. That's why it is coiled right-handedly. So hydrogen bond is must in secondary structure. So this is present in the single strand. But in beta pleated seed, you can see that there are two polypeptide chain which are joined with the help of hydrogen bond. So this is beta pleated seed. Similarly, in collagen, three such chains are there and they are joined by hydrogen bond. Then come to tertiary structure of protein. The overall three-dimensional structure of a polypeptide is called its a tertiary structure. Very, very important. And I have told you that the tertiary structure is due to the bond formed between the R groups, not between the NH or COOH group. It is due to the formation of the interaction between the R group. So here you can see that this is a polypeptide chain, and how the R groups of amino acids are linked with one another. So here we can see various types of bond, as I have shown you. Here you can see that this is your ionic bond in between NH3 and COOH. Here is ionic bond. Then here you can see hydrophobic interaction. Then here you can see disulfide linkage, hydrogen bond. So all different types of bonds are present between the R groups of the. So here you can see that this is the R chain. Okay, the variant group. This is also the variant group is linked with the other amino acid. So this is also variant group because here CH2 is there. So from that it should be clear to you. So this is also a variant group. This is also a variant group. This is also a variant group. Excuse me. So in this way, the tertiary structure is much folded and it provides a three-dimensional structure. Okay, and it is more complex and it is the functional form of or active form of the protein. Okay, so here you can see how by the formation of these bonds, the 3D structure of that is the protein is obtained. So this is the three-dimensional structure formed by the formation of various types of bond, that is hydrogen, disulfide, hydrophobic, ionic bond, other than that is along with peptide bond. Then the last one is quaternary structure. This is made up of more than one uh, polypeptide chain, and each polypeptide chain is called subunit. So here each polypeptide chain is called a subunit, and when all these subunits come together, they give the protein a quaternary structure. For example, human hemoglobin consists of 4 polypeptide, means 4 subunits, and out of which 2 are alpha type and 2 are beta type. So, in hemoglobin, you can see that this is hemoglobin, structure of hemoglobin, and in hemoglobin you can see that these two are beta chain and these two are identical, that is alpha chain. Okay, so there are altogether 4 subunits are there, and all of them are attached to the hem group, that is iron. That's why it is called hemoglobin. So it is the most complex structure, and you know the function of hemoglobin, it carries oxygen. But when you compare it with collagen, so in collagen also when they form pleated structure, or the collagen may show secondary structure as well as quaternary structure. In secondary structure, they lie side by side and they form the pleated structure. But in quaternary structure, the 3 polypeptide chains are joined in this way and they form a quaternary structure of the protein. Okay. So here again the comparison of all the 4 types of proteins I have given. So primary one is very simple one, secondary one is of 2 types, one is a pleated sheath and other one is alpha helix. Alpha helix consists of only one strand, one polypeptide chain. Pleated sheath consists of 2 polypeptide chain, and collagen consists of 3 polypeptide chain. Then tertiary one, single polypeptide chain is folded upon itself to form a 3D structure. Then quaternary consists of more than one polypeptide chain which are combined together with the help of certain types of bond. Now we'll discuss some other functions of a protein which we have already discussed some of the functions, but these are the some other functions. The enzymes are proteins that facilitates biochemical reaction. For example, pepsin is digestive enzyme, and trypsin also we have studied. Antibodies are also protein, that I have already told you. DNA, the genetic material, that is also associated with protein and that regulate the structure of chromosome. Then the contractile protein, actin and myosin are present in the muscle and helps in contraction and relaxation of the muscle. The structural protein provides structure and support to our body. For example, the collagen and elastin are the present in the connective tissue. Then some hormones are protein in nature and they coordinate various functions in our body. For example, insulin is a hormone, protein hormone that regulates the blood sugar. Then transport protein which are present in the membrane and as well as they are present in the other parts like connective tissues. For example, hemoglobin is a transport protein which transports oxygen through the blood. So this is all about the protein and amino acid, children. So I hope I tried my level best to clarify all the concept related to amino acid and proteins as well as to the biomolecules. So in our next session, we'll discuss other 2 biomolecules, that is carbohydrates and lipids. So thank you very much for watching the video till end. So children, wherever you have any confusion, just pause the video and listen to me whatever I am saying, whatever I am explaining, so that all your concept will get clarified. Thank you one again. Thank you very much, children.